We are looking at 48.4 and how neurons communicate with one another through synapses. And we're going to look really quickly at electrical synapses and then spend the rest of our time looking at chemical synapses. And so electrical synapses are ones that send electric currents. So that makes a lot of sense. The message is going to be sent more quickly because they're sent through gap junctions from one neuron to another. These are used for rapid, consistent movements, like your heart beating or peristalsis, so that's your food moving down your esophagus and through your GI tract. Also for fish flipping their tails to stay in the school, and so they have to swim and change direction very quickly. So that's pretty cool. And so that is pretty much it for electrical neurons. We're going to now talk about electrical synapses. And so now we're going to talk about chemical synapses. And that's the one that we have spent the most time talking about already. And so while a neuron is at rest, what it's doing is it's trying to sort of regain itself. It's trying to pack these synaptic vesicles with neurotransmitter. And so these synaptic vesicles are going to hang out in the synaptic terminal. So here is our cell body, and here's the axon, and here is the synaptic terminal. And here's the membrane right here. And so we can see that this is the postsynaptic neuron here. And so when an action potential arrives, as it travels down the axon, when it arrives to the synaptic terminal, it's going to trigger the calcium-gated ion channels to open, which is going to allow calcium to flow into the cell. And so calcium, what it's going to do is it's going to break bonds they're not shown in this picture, but these vesicles are bonded to the membrane, and so it's going to break those bonds, and it's going to allow the vesicles to form with the membrane, and it's going to, through exocytosis, it's going to dump out those neurotransmitters into the synapse. Those neurotransmitters are going to pass over the synapse into the postsynaptic membrane, and it's going to be received there. So here, when the neurotransmitter binds to the other side, it's going to be binding to a ligand-gated ion channel, basically a neurotransmitter gated channel. And so what it does is it allows different ions through. So it's going to be the neurotransmitter that bonds to the gate, but the gate is opened to ions. So if it lets in some positive ions like sodium or potassium, what that's going to do is that's going to depolarize our cell. It's going to make it a little bit more positive. And so that's going to bring us closer to the threshold potential. So causing depolarization. Think EPSP, excitatory postsynaptic potential. This means that it is sort of exciting the nerve, and it's going to potentially cause an action potential. And on the other side of that, an inhibitory postsynaptic potential, or IPSP, that's if K plus moves out of the cell. So in the postsynaptic cell, if the positive energy is moving, or the positive ion is moving out, or the negative chlorine moves in, what that's going to do is that's going to bring the neuron lower. It's going to hyperpolarize, and it's going to make it more difficult for the cell to reach that threshold potential. So this is an inhibitory postsynaptic potential, and it's causing hyperpolarization. So when we study neurons, we think about a single synapse. However, in reality, it is hundreds of synapses. Here, let me play this video for you. These are the, I have to keep it going. These are the neurons of a mouse's brain. And so the yellow is going to be the actual neurons, and then the magenta and the teal are the proteins at the terminal ends. And so why do I show you this? I show you this because it's just more complex than one neuron meeting one neuron at a synapse. It's many terminals and many presynaptic and postsynaptic terminals. So I want you to keep that in mind because there's something called summation. So if there are multiple potentials or multiple ion potentials happening all at once, they're going to be summed together. Let me explain that a little bit more clearly on the next page here. So a summation, think wave construction or wave destruction. So when you have the positive ions coming into the neuron or through the membrane, it's going to cause a sort of wave, like a graded potential. And so here, let's talk a little bit about physics. Let's think of waves for a second. Wave construction and wave destruction. Wave construction is when you add two waves together. So if you were to be one person out on a slinky, is one person on one side of a slinky over here and one person on the other side of the slinky over here. If they are both grabbing one end of the slinky and they're going to cause a wave, those waves are going to move towards each other. And so let's say that wave on this side is a half a foot. And let's say that this wave on this side is a half a foot. They're going to move towards each other. And when they are together, we're going to find their sum. They're going to be added. And so that's going to be one whole foot. And so we call that summation. But we have to also think about wave destruction. Wave destruction is the opposite. So if you have this person on this side making a positive wave going this way, and then this person on this side is making a negative wave, so it's coming in this way, it's going to come together, and what they're going to do is they're going to cancel each other out. So we have 0 feet. And so this is the same thing that's going to happen with our potentials in the neuron as they move towards the axon hillock. And so if we have a summation, there's two types of summations, temporal and spatial. So if we have a temporal summation, what happens is we're going to have multiple waves or multiple potentials interacting. So let's think about a slow tempo. And so we think temporal, tempo, think music, tempo. And if we have a slow tempo, here, 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 what happens is those potentials have a chance to increase and then decrease back down to the resting potential. And then it happens again. It has a chance to increase and then decrease. So there's no summation happening. There's no wave construction happening. But if you're to increase the tempo, then you have one wave. And before it has a chance to go back down to resting potential, it's going to increase again. And so we're going to sum up those waves, and it's going to increase, and it's going to increase. And it's when you have those very large waves or those very large potentials that reach the axon hillock, that's going to create your action potential. So we can have this with a fast tempo or temporal summation, but we can also have a spatial summation, and that is just if you were to have, let's say, let's call these your presynaptic cells. Let's call this your presynaptic cell. So if you have lots of neurotransmitter coming and hitting at all angles, and then they interact inside the nerve, so inside that postsynaptic cell, they're going to sum up, they're going to interact with one another, and they're going to create a large wave, which is going to more likely cause an action potential when it reaches the axon hillock. And so that is summation. And so after a signal is fired over a synapse, it needs to return to a resting state, and the neurotransmitters need to go. One or two things are going to happen to it. One is that it's going to they're going to go be incorporated back into the vesicles and used again, or they're going to be inactivated by an enzyme, basically just hydrolyzed and moved off and used as something else or potentially reconstructed again. This clearing up process is very important because it allows for the the nerve to fire again. And if you don't clear it up, then it can't fire again. And so, an example of something going wrong, of science going bad here, is sarin gas. This is a chemical of mass destruction, and what it does is it releases it releases acetylcholine into the synapse, and acetylcholine needs to be broken down by these enzymes and removed. But what sarin gas does is it inactivates these enzymes, so the acetylcholine isn't cleared out. And so the muscles in your body they're skeletal muscles will tense or flex, and they won't unflex, and they'll flex some more, and they'll flex some more, and it'll increase. And then what happens is your body, your entire body just starts flexing, including like your pupils, your heart, your diaphragm, which makes it so that you can't breathe and you eventually asphyxiate. But so it's awful, a horrible way to go, and that is why it is not allowed to be used even in war. But that is, oh, where did it go? But that is sarin gas, which is a nerve gas. Acetylcholine. So we were just talking about acetylcholine with the sarin gas. It is a neurotransmitter that which includes muscle stimulation, memory formation, and it's incorporated in learning. There are two major classes of acetylcholine. Ones that are broken down by an enzyme, and another that is used when it's broken down, there's a signal transduction pathway. However, do not worry about the signal transduction pathway. That's going to be for your heart, for your central nervous system, and we haven't talked about signal transduction pathways. And so don't worry about that. That's the part that I told you not to worry about. So some chemicals can mimic or alter the effects of acetylcholine. And an example of that is nicotine. So it acts as a stimulant. It does exactly what acetylcholine does. It's an excitatory. It's a stimulator, and it's going to mimic the neurotransmitter by bonding to the receptors. So it fits into the receptors. And so the body thinks that it's receiving acetylcholine. And so it's going to produce the same stimulations, the same memory forming, and botulism is another example of something that can go wrong with acetylcholine. So it's kind of the opposite to sarin gas. Botulism inhibits the release of acetylcholine, so that makes the muscles continuously contract. So it's the opposite of sarin gas because sarin gas didn't allow for the neurotransmitter to be removed or the acetylcholine to be removed. And so the muscle flexed. And with botulism, there is no neurotransmitter being released, and so the muscle contracts. And so it's not a pleasant thing to experience. And people actually, so Botox treatment, this is what people they put into their lips or around their eyes or here on their forehead. And so it inhibits or it stops the muscles from not flexing. And so it makes it look like there's no wrinkles. It fills up that space. So you're actually putting a toxin in your body to get rid of wrinkles. So amino acids are a type of neurotransmitter, or we use amino acids as a neurotransmitter. And so glutamate is one of the most abundant neurotransmitters in the central nervous system and necessary for the formation of long-term memories. Another important thing that we're going to be talking about is GABA. This increases the memory's permeability to chlorine, which will create an IPSP, a hyperpolarization. So if we think back to when we were talking about how if we have chlorine, let's see if I can find that here. If we include chlorine into the postsynaptic cell, it's going to lower it's going to hyperpolarize the cell, moving it further from threshold potential. And so it's going to make it harder for the cell to reach action potential. So fewer neurons are going to be firing. And this is going to create a less anxious state. This is going to create the opposite of excitatory. It's going to be an inhibitor. And so this is essentially what Valium is. It decreases anxiety. You're firing fewer neurons, so you're going to be less anxious. So this is GABA. The next form of neurotransmitters that we're going to talk about are biogenic amines. Amines, think amino acids. These are synthesized from amino acids. First one, epinephrine, is correlated to tyrosine. And it's also known as adrenaline. It functions as a hormone. It acts as a hormone because we call it a hormone because it's a neurotransmitter it's not created in the brain. So therefore we call it a hormone. But what it does is when it's released in your in a gland above your kidneys. And when it's released, what it does is it's an excitatory hormone, and it's going to cause your pupils to dilate. It's going to cause your lungs to expand so you can take in more air or more oxygen. It's going to cause your heart to beat faster. It's going to redistribute blood to your muscles and maximize your glucose blood levels. Think escape from a predator. That is essentially what epinephrine is made to do. It's to help you survive. So when something startles you, your hair stands on end. It makes you look bigger to scare off a predator. This is epinephrine, also known as adrenaline. Norepinephrine, also interacting with tyrosine, is an excitatory neuron which also functions as a hormone. They're similar, they're not exactly the same, but they're close enough that we're going to leave it at that. Dopamine and serotonin, they get the title of being like your happy hormones or your happy neurotransmitters. And what they do is they affect sleep and mood and attention and learning. And so oftentimes depression is treated by enhancing the effects of this serotonin by inhibiting the reuptake of it. So it's not going to be taken back into the presynaptic nerve yet. It's going to spend more time being taken up by the postsynaptic nerve. So it's going to have a greater effect. And so that is one way that we can treat depression. Neuropeptides, these are short chains of amino acids. An example of neuropeptides are our endorphins. And so these act as an analgesic, which decrease pain. Like sometimes when we take a pain reliever medication, those are analgesics, and they decrease pain perception. They can also reduce urine output, decrease respiration, but most importantly, they can produce euphoria. And so your body is going to naturally, if you're a female, your body's going to naturally produce and release endorphins during childbirth. And after you exercise, you feel great. You feel like you can take over the world because your body is releasing endorphins. You have a sense of euphoria. And so opiates, which are a real problem in our world today, heroin and morphine, what they do is they bind to the same receptors as endorphins, and they produce the same physiological effect, but sometimes at a much greater rate than what your body would be producing. And so therefore, they can have negative effects. But that's all an opiate is it just mimics an endorphin. Finally, the last one. Our body uses gases like nitric oxide to act as a neurotransmitter. And an example of this, nitric oxide is released into erectile tissue in the penis during an erection. So what it does is it goes into the muscle in the penis, and it makes it more spongy, and it allows for room for blood to rush in and to cause the erection. Viagra, known as the little blue pill, works to allow for an erection to last longer. And what it does is it works by stopping the enzyme that stops the nitric acid from making the tissue spongy. So basically, it allows the tissue to be spongy longer so that the erection can last longer. And so that's it. That's 48.4. And if you have any questions, please let me know. Have a great day.